Okay, welcome everyone, and good afternoon to all of you. It's my privilege to invite all of you to attend this first Computing for Global Challenges Symposium. Symposium marks the culmination of an 8-week program that was conducted as a part of C4GC's initiative that we started here at the Biocomplexity Institute. And the program aims to train undergraduate students in the use of computational methods to address global challenges pertaining to human health, infrastructure, and environment. During these 8 weeks, students worked with faculty members on ongoing research projects. The work and their day-to-day interactions taught them the process of doing team science and to do cutting-edge research. They did this in the context of solving real-world problems. We embraced a team science approach because these problems require a multiple set of views and multiple set of skills. In the process of working on these large-scale problems, students learned about cutting-edge software systems, but also mathematical modeling, state-of-the-art methods in machine learning, computational biology, agent-based simulations, and data science. The first year of this unique program had 13 undergraduate students participate, and they were chosen from a very strong pool of candidates who applied. We had most of the students from UVA, excepting one who is going to go to Rutgers as a first-year student starting in fall. But we hope to have students from other universities join us as well in the coming years. Most of you are from the computer science department and mathematics department, but again, this was something we started somewhat late. We hope that others from other departments will also join us in the coming years. As you all will hear, the students have done an outstanding job. I'm really looking forward to the presentations and the poster session that follows. And I hope that many of you will return back to BII and NSAC to do further research and possibly even to do a PhD with us. Programs such as these can never be successful without the help of a number of individuals. And it's certainly not something that one person in the division or in the institute has done. So I want to thank all the faculty and staff in the institute and the division for helping us make this into a successful program. I do want to call out a few individuals who have played a very, very important role and gone beyond and above what it takes to do their day-to-day work. And they include Erin, Samarth, Abhijin, Andrew, Lorna, Jill, Grace, Beth, Ravi, Brian, Jennifer, Keshawn, Scott, and Michelle as well. And if I missed somebody's name, I apologize. This is the names that I sort of figured out as time went by, and I saw folks involved. But in general, I felt that everybody at the institute here was helpful, helped the students with questions, and guided them as time went on. From simple things like having a pizza party for the students that Erin and Ravi organized, to having a lock-picking exercise, to ice cream trucks, to other exercises and fun-filled activities. I think you folks hopefully enjoyed these 8 weeks that you are here. I also want to thank all the mentors. This program certainly cannot happen without your mentorship. And I know that it's a time-consuming process, but it comes with a great sense of satisfaction. And there's no better sense of joy to see one of your students complete the work well and excel in doing so. And I'm sure that when one of these students look back at this program 10 years from now, they will find this a very fruitful experience, and many of them might point to this program as a turning point in their career where they decide to take research as a career path. So I want to thank the mentors again. And finally, I want to thank Chris and Laurie. Thank you so much for helping us. And thanks to Kevin Scadron in the CS department, who was very helpful, as well as Sally, whose program in the Northern Capital Region played an influential role in us establishing this program. And a final thanks to our sponsors, NSF, UVA, NIH, DoD, NASA, and many other funding agencies who have funded many of the scholarships you folks got. So thank you very much. I look forward to the fun-filled afternoon, and hopefully you'll enjoy the rest of the 2 days and the symposium.
Hi, I'm Miranda. My project for Computing for Global Challenges was to conduct a literature review on quorum sensing, and particularly quorum sensing as modeled through computational means. So just some background. Quorum sensing is a mechanism by which bacteria decide whether or not to engage in collective behavior. And they do this by secreting small chemicals called autoinducers that accumulate in the extracellular environment. And just as sort of an example system, the bacteria Vibrio fischeri uses quorum sensing to regulate bioluminescence in its squid host, the Hawaiian bobtail squid. And as you can see sort of at the top there, when Vibrio fischeri is at a low cell density in the squid, it does not bioluminesce, because if there are only like 1 or 2 bacteria that are engaging in bioluminescence, it's not going to light up the whole squid. And therefore it's not worth it for the bacteria to invest in producing the enzymes required for bioluminescence. But once a certain cell density is achieved, as you can see in the diagram there, autoinducer accumulates outside the cell, it gets imported into the cell, it binds with a receptor that then upregulates transcription of the luminescence genes. And particularly important are LuxA and LuxB, which then combine to produce luciferase, which is the main enzyme involved in bioluminescence. And so, therefore, at a high cell density, all of the bacteria, at least in theory, are bioluminescing and the whole squid lights up. So this isn't the only behavior that can be regulated by quorum sensing. Biofilm formation, exoenzyme secretion, virulence factor production, all of those things can be regulated by quorum sensing. However, any sort of cooperative behavior like that comes with the problem of free riders. So one thing that we were looking at is, why is cooperation worth it when, say, a bacterium that doesn't bioluminesce at all can still live in the squid and derive the benefits without also paying the cost of producing luciferase? So some different mechanisms by which bacteria can prevent cheats from overriding the population are through kin or kind discrimination, where the bacteria recognize other bacteria that are cooperating and make sure they only direct the benefits to other cooperating bacteria. And that could manifest, for example, through a punishment system. For Vibrio fischeri, we think that the squid itself actually punishes bacteria that aren't cooperating, but other bacteria are sometimes able to punish each other, maybe through their quorum sensing molecule is a toxin, and part of the gene that's being upregulated is the antidote to that toxin. And if the cheating bacteria don't respond, then they're killed off. Or maybe cheaters are intrinsically better suited to survival and will increase in proportion in the population. But because bacterial populations are disturbed so much and so frequently new bacterial populations are forming, bacterial populations that are just cooperators may be able to grow larger by virtue of the fact that they're cooperating and offset other populations in which cheats are overriding those cooperators. And that's called Simpson's paradox. But anyway, the end goal of this project was to develop a common minimal model that would allow us to see how quorum sensing emerges as an adaptation in bacteria where previously cooperation was not necessarily occurring. So in our design, and not all of it is in this model up here, but we have 6 states, and 4 of those states are sort of basic actions that any bacteria would perform, like moving or dividing or dying or just absorbing nutrients. But we also worked in a cheap extracellular compound, which does have a small benefit to the cell, but can be produced without much cost to the cell as sort of a potential molecule that would become an autoinducer theoretically in a real-life population. And then also an expensive extracellular compound that would act as our public good, where it's very difficult for the bacteria metabolically to produce it, but as long as it stays around the cell in a high enough concentration, or other bacteria are producing it in a high enough concentration, it can still provide benefits to the cell, and higher benefits than just the cheap extracellular compound would as the autoinducer. That's all. Oh, wait, sorry. And these are references for this PowerPoint. Those weren't all of the references, or all. There certainly is not a comprehensive list of the literature that I reviewed, but that was the information relevant to this presentation. But.
Hi all. My name is Daniel Wang, and so my project for computing for global challenges is a network analysis with Twitter data. And alright, so during my project, there are mainly three parts. The first part is the data collection. But first, I want to introduce the background of this project. So the background of this project is first that it is to complement with UVA's effort to participate in the Social Sim Challenge sponsored by DARPA. So basically, the Social Sim Challenge is a challenge to simulate Twitter behaviors and to predict user behaviors as well. So and also just to the public interest, so there's a lot of news with regard to social media recently. So there's data breaches and other scandals. So this work will be to help predict some of the and to stop some of the behaviors that currently exist on social media sites. So to make a successful effort to collect the data, we have to make a tool for that. The reason is because that Twitter has multiple API limits that such as that you can only see certain amount of followers per user, and you can see certain amount of tweets per user. So in order to optimize our effort to extract the most amount of data from Twitter, we would have to build such a data collection tool. All right, so the foundation of the data collection is built on the followers network. So here's the structure for it. So basically, in order to construct a followers network, again, there are multiple networks that exist on Twitter, such as followers network. There's also mentions network, so based on network constructed based on hashtags, on ad symbols or based on hashtags. And the follower network is constructed by, so you will start with a root user and see, and all the edges are constructed by if they are following someone. So this is the structure. Obviously, you see from a root user, you will go through a portion of his followers, and you will expand the top 10 followers based on how active they are, and to repeat that process in order to pull enough users into the network to construct a large enough network. And now, in that process, there are some issues, such as the most active users. They may be bots. They may be automatically programmed to retweets and tweets a large chunk of data. So that's why I also integrated a bot detection algorithm to filter out these bots in order to make a more robust network. And after the construction of the network is done, we'll see each user's Twitter timeline, and so the timelines consists of all the users' history, their tweets, retweets, and all their interactions with other users. And for the result of the data collection, we have collected 20 million tweets. 12 gigabytes of Twitter data and 3,200 tweets per user, if, which is the Twitter's API limit. Yes. Next slide. All right, so after the network is constructed, we also make a semantic network for each user with two tools. The first is Spacey, which is a natural language processing tool utilized to tokenize users' tweets and into words and recognize those words, they're parts of speech. And the second tool we utilize is NetworkX, which is a tool to construct a network based on all those words turning into nodes in the network. And after we construct a network for each user, we'll make several visualizations. As you can see, they are done with the tool called Gephi. So the first one is the user network of the Twitter data I collected. So it consists of roughly 9,000 users, and each edge will represent if a user is following the other user, that there will be edge between those two users. And the second graph over here is the semantic network for user, just for example. So the size of the words denotes how frequent those words exist within that user's timeline, and within each tweet, each words in that tweet will be connected with each other words in the tweets with edges. And lastly, it's just a word cloud for that user, and obviously the larger size denote that those words appear more frequent in that user's timeline. And the last thing we did is to make a user behavior prediction tool. So the goal of this is to predict what user might do when this user see a tweet. So those are within Twitter, those are the possible user interactions they may have with that tweet. So this user may retweet that tweet when the user sees that tweet, or reply, or quote, or like, or maybe when the user sees that tweet, the user does nothing, so it will be no action. And in this process, the parameters are for centrality measures. So the centrality measures are basically measures that to determine how important vertices are in a graph. And this one is done by first tokenize the text and to calculate the individual centrality measures of the text of the tweet, and to add up all the to sum up all the centrality measures of all those words in a tweet to construct a training data set. And also the training data set also contain features from the user level data. For example, it might be helpful to know that this user is mostly likely to retweet another user who may have a lot of original tweets, and it could be helpful to know. So that's why I included that into our training data set as well. And for the results, so the accuracy of a multi-class classification. So basically, my model intend to produce a probability measure for each of those 5 actions that the user may take when they see a tweet. And the accuracy is around 57% for multi-class classification. And here you can see the RC curve for that. So obviously obviously my model currently is better at predicting some users' behaviors. I believe it is more likely to predict if the user likes something or the user retweets something. And so here's a graph of so basically I will have to write a series of scripts and to connect them together in order to produce the output. All right, so for the future improvements for my model, I will obviously need a larger data set to test about the accuracies, and also I will intend to do a deeper analysis of the correlations of the features. So to filter out some of the features that's less relevant in order to make a more robust model for this tool to make predictions. And thank you.
All right. Hello, I'm Ethan. My project was on mapping several invasive plant species using deep learning in a region in Nepal. So the objective of this project was basically to examine the spread of various invasive plant species and then to analyze the impact that human activity and climate change has on this spread. For this research project, we focused our study on the Chitwan Annapurna landscape in Nepal, just because it was a biodiversity hotspot, so it had a lot of factors that we could look at. Our general approach for this project was to utilize a machine learning algorithm to identify the presence or absence of a specific plant in a satellite image, given like the image data, and as well as several like ecological factors such as like temperature, slope, and elevation. And so for this, we trained a deep learning model using multispectral data and survey data that was obtained through collaboration with the Tribhuvan University in Nepal. So the data we were given for this consisted of multispectral satellite data from the Digital Globe Worldview 2 satellite, as well as survey data taken in Nepal. So the multispectral data contained image data over 8 several bands. So and then the survey data taken in Nepal contained various like ecological factors, like I said, like elevation, temperature, and slope, as well as the presence or absence of 7 different plant species. And so what we did was we combined the multispectral image and the presence and absence data from the survey to create a data set of labeled images. And we used this to train a convolutional neural network, which once trained could be used to predict the absence or presence of a specific plant over an entire satellite image. And then once we have those results, we can map it onto satellite images from varying time frames so that we can see the spread of this plant as time goes on. So for this convolutional neural network, we based the initial model off of examples from the Keras documentation, and we just generally improved on it by using our our own data set. Our final model had 4 convolutional layers and 3 fully connected layers. But in training this, we ran into a bunch of challenges. For one, the way that the survey data was taken, they basically had a device that gave them the longitude and latitude points of the point that they were standing at. But what they did was they just kind of looked around to see if this plant was in the area. So there was like, I guess, an error of around like 10 meters of inaccuracy of where the latitude and longitude points pointed to and where the plant actually was. So we had to choose like an image size that took this into account and could correct this. Additionally, the data set was relatively expensive and focused mainly on roadside areas. So this helped if we were only looking at the spread along the roadside, as we would need less images. But for our model, when we tested it across like the entire image, it was much less accurate in areas that we did not train for, such as like deep forests. And as previously mentioned, we had insufficient data. As the plant that we had the most data for, Lantana, we only had 61 points. And of those 61, we only had 14 which showed presence. So we had to introduce a lot of artificial samples through data augmentation techniques so that we could properly train this them. So our best model was able to relatively consistently get around 95% training accuracy and around 85% validation accuracy. So we used this model to predict Lantana on the Tanahan district, which is the image shown on the left. And then once we got these predictions, we mapped it onto the original image, which is shown on the right, where the green shows the presence of the plant and the red is the absence. And then so now we're currently in the process of obtaining more images and more survey data so that we can evaluate or more accurately evaluate how accurate our model is and then go from there.
Hi, I'm Anna Brower, and this summer I've been working on the vulnerability in the context of Hurricane Harvey. So what I'm trying to do is prove upon the current measures of vulnerability by looking at what tools are currently available and seeing if these are a good indicator of the hazards that come from hurricanes. And I'm looking particularly at Hurricane Harvey, which was one of the most catastrophic and costly hurricanes in history. So Hurricane Harvey started in late August 2017, and it was a Category 4 hurricane which caused about $125 billion in damage, and it made landfall about three times in 6 days, which caused a lot of flooding in the Houston area. There's many areas in the Houston area that experienced about more than 50 inches of rain, as well as 5 feet of flooding, which forced thousands to evacuate. And then, so one of the current tools to assess the vulnerability of populations is Cdc Social Vulnerability Index, and this is a ranking which are called the RPL themes. And these RPL themes quantify the vulnerability of each tract in this area based on a combination of different variables. So there's one overall vulnerability ranking which is based on 4 different RPL themes, which are socioeconomic status, household composition and disability, housing and transportation, and then also minority status and language. And these different rankings are made of a bunch of different factors, like how many people have vehicles in the area, or like how many people are above 65 or younger than 17. And then, so these rankings are valued from 0 to 1, where the greater values indicate greater vulnerability. And then on this top visualization I made, this just shows the overall vulnerability ranking for each of the tracks, and the darker colors show the greater vulnerability. And then this next visualization on the bottom shows the inundation extent from Hurricane Harvey, where the red area shows where the flooding was present. And then after I created the visualizations and looked at the data, I did some statistical analysis. And what I wanted to figure out when I was doing this is whether there's a relationship between the percentage of inundation and the different RPL themes. So what I did for this, first I looked at to see if there's spatial autocorrelation present in the RPL themes. And to do this, I used Moran's I, which so that measures spatial autocorrelation, or how related the variables are based on their location. And then this is valued from -1 to 1, where the -1 indicates that the data is perfectly dispersed, 0 randomly dispersed, and then 1 means the data is perfectly clustered. And from this, based on this calculation, I found that in all of the RPL themes, there's a significant amount of clustering. And then from there, what I looked to see, I created a regression line based on the percentage of inundation in all of the tracks and the different RPL themes. And from this, I found that there is not really a relationship in the currently the vulnerability is assessed in the population, and between that and the inundation. And as well, there's still clustering present in the data. So that's all.
Right, my name is Tim, and my project was on the CRIS 2.0 disaster evacuation model graph generation. So CRIS 2.0 is a project funded by the NSF that deals with evaluation of infrastructure, and to be specific, CRIS would stand for Critical, Resilient, Interdependent Infrastructure Systems and Processes. So some background on my project is related somewhat to Anna's project. The concept that hurricanes and other disasters can cause damage to a region, and it's important to understand how people behave, what kind of infrastructure we have to support these kind of evacuations, and in terms of my contribution, to create a model that would assist in the visualization and understanding of these kinds of disasters. So my project dealt with the generation of graphs to represent neighborhoods and to simulate how people would evacuate. So some concepts that we need to introduce are this directed graph, which is based on Kleinberg small world networks. And so we can view nodes as being families or homes, and we can view edges as influences that families have on each other. So in a real world context, this would be something like one family chooses to evacuate, and then a neighbor would see this family evacuate and would therefore choose to evacuate as well. The edges within this graph consist of short-range edges, which simulate neighbors. So people within a certain geographic distance threshold of one another would be influenced by their neighbors, and long-range edges, which represent more geographically distant friends of the families who would nonetheless still have influence. And to simulate clustering in these networks, we utilize a distance-based weight system, which is used for long-range edge selection. So my goal in this project was to implement multi-threading and parallelism in the project to accelerate the runtime of this graph generation. Inputs included the node data, such as the latitude and longitude coordinates, the number of threads desired for the run, the distance thresholds, which determine short and long-range edges, and random number generation to assist in multiple runs of the code. And so the structure of our code was a nested for loop to assign every node its neighbors, and my parallelism was intended to accelerate this process. So some results of the runtime indicate that the use of more threadeds resulted in a moderate increase in time. As more nodes are added to the graph, the runtime increases exponentially, and we expect a greater benefit when this is run on more massive scale graphs. So some future work that could be done includes further testing on different machines, a distributed system which could potentially parallelize the project even more by assigning partitions of the nodes to multiple machines, and implementation of faster searching methods, such as binary search in the long-range edge selection process. Thank you.
Good afternoon. My name is Henry Karskedden, and I was working with Dr. Chen on forecasting time series data with influenza. So rather than forecasting the traditional case counts, we were forecasting CDC data that ranked the flu in intensity levels from 1 being the lowest to 10 the highest. So this is ordinal discrete data rather than more continuous data. And so this was available for 50 states on a week-by-week basis. On the side here, you can see a little plot of the states for the 2017 to 2018 season and the 2018 to 2019 season. And so this region right here is where they're more likely to fit in, but as you can see, there's a lot of variability between states. So sort of focused on trying to model a good fit for each state. Another interesting feature of the data would be the high amount of autocorrelation in it. So this informed our modeling a lot. And as you can see here, this is a partial autocorrelation plot for Virginia, which is sort of representative of the other states. Most of it is due to the lag 1 autocorrelation. So that's the most recent time steps, the most recent observation. So there's a little bit of past modeling that had been done that I was working on improving. It was an ensemble method that used the SEER system of ODEs, an ordinary regression on the time series, in addition to this naive method that I defined below because it's sort of a more unusual one, and a Markov chaining model. And so interestingly, the ensemble and all of these more sophisticated models were vastly underperforming this naive method. And so I set about trying to find some new models that might improve the ensemble to the point where it's passing this benchmark that we set here with naive. So started off by exploring recurrent neural networks. And so if you're not familiar with them, they're pretty similar to your normal neural network, except for they're intended for sequence data. So naturally, it might be a good fit for a time series. But because we're relying on a univariate time series, and there are a lot of parameters to be tuned with not too many training examples, they didn't fit the data very well. So moved on past those. Then we took a look at some more traditional machine learning methods, such as various ensembles of decision trees, as well as support vector machines. Support vector machines actually turned out to be quite a good fit for the task, whereas the decision tree ensembles didn't do quite so well. Not entirely sure why they were performing so poorly. Also looked at some classical time series models, such as SREMA. So that's the seasonal variant of ARIMA, which is making use of differencing and autoregression to pull out some of the autocorrelation in that data. And also Holt-Winters, which also makes use of the seasonal structure of the data, tries to parameterize the model to fit the structure of the data. So, moving on, these new models that were performing quite well still weren't bringing the performance of the ensemble up to anywhere near the benchmark. It sort of stalled out. And so we figured out that this is because it wasn't really taking into account the accuracy of the base classifiers very well. So what I did is I set about trying to create a different mechanism to ensemble the base classifiers and use that accuracy information going down to the weekly and state level. So that actually ended up improving things a lot when I got rid of the Sierra model, which is performing quite poorly, added in the support vectors and the two classical time series models that I discussed earlier. But as you can see down here in this table, the performance was not unequivocally better for this new ensemble. And in fact, the naive actually did significantly better with this Matthews correlation coefficient, which is sort of a providing a balanced interpretation of the confusion matrix. And so, in my opinion, looking at this, it seems to be that devote some further exploration to the SREMA, which on all three of these metrics sort of sits towards the better end of performance. And so there's some possible room for improvement there by pulling in some outside data, such as climatic data for each state. And it's also worth noting that the performance for all of these methods was the poorest for the classes that were traveling between the valleys to the peaks, and between the peaks to the valleys, because there's the most changes in slope and variation from that. So that concludes my presentation.
My name is Katarina Kemper, and my project is human immune cell responses to bacterial pathogens. So our immune system is basically there to fight bacteria and anything bad, and bacterial pathogens are like the bad bacteria. And the immune system has something called the macrophages, and they're basically there to engulf the bad bacteria, and it creates like, the macrophages are here, and then this infection or this bacteria comes in and they fight, and it creates this huge battle. So what Nedelac looked at was when this fight happens, this huge battle, what genes are being expressed, what is happening in this macrophage to fight back this bad bacteria. And so what they used was this RNA sequencing, which you can see right here. And the RNA sequencing basically has this RNA sample, and then uses the cDNA library and creates these reads, which are these long DNA fragments. And then we align it to a reference genome right here. You can see that there are a bunch of like reads, and then we have these genes, and that's how we get like the number of genes and how many times they're expressed based off these long DNA fragments. So what my project is, we used RNA sequencing to see if when there's two battles. There is when a macrophage and listeria come together, what immune responses we get, and then the other battle is when a macrophage and a salmonella comes together, and then we compare them and see if there's similarities or differences. So what we did is we grabbed a bunch of data from the sequence reference archive, and then we did RNA sequencing it by referencing it to a human reference genome, the GRCH38, and then we looked at which genes are the most expressed. And the level of significance was 16, which means it had a log 2 fold change of 4. And then we ranked them of how significant they were. And so in this battle, these genes are basically like the soldiers, and the most significant ones are the ones that have like the most stars, or like the super soldiers. And we wanted to see which ones are the super soldiers, and then see how many of them there are. So we wanted to visualize it and see compared to the total genes that are being expressed. And then we wanted to look at their functions, and if between Salmonella and Listeria, if they have certain functions that are the same. So this is our volcano plots, and volcano plots are basically giant scatter plots to look at drastic changes in the log 2 fold changes. So you see this is this right here is the Listeria, and the green dots are the most significant ones, the super soldiers. And we found that 0.79% of all the total gene are the super soldiers, but Salmonella has 2.64%. That's so much more than this Listeria had. And then we also looked at if the Listeria and Salmonella had genes that were the same, and we found that 233, and these are the purple ones right there. Okay, and then we also looked at the data themselves. So we have Salmonella on the left and Listeria on the right, and we found we can see the top 3 with the highest log 2 fold changes, which means they were most highly expressed. And Salmonella had the most expressed log fold change with 11.28. And you can see that they have the same number 1 gene, which is IL-12B. And then, so this could be like the main gene that's like the most expressed. And we also looked at the molecular function using a gene ontology. And you can see like there's white boxes and like darker orange, and those are how important they are based off their function. Also like the cytokine signaling function. Cytokines are basically the messengers. So in a battle, there are people that have to talk to each other. So the genes, they talk to each other, and they use cytokines for that. And if you look at the red boxes, those are the most significant ones, and we see that the molecular function for both of these, it's a little small to read, but it's ion binding. So for Listeria and Salmonella, the general antibacterial response is ion binding, and for Salmonella there's also another red box, and it's for DNA binding. We're still looking if there's other diseases, if they also share that molecular function.
How's it going? My name's Kieran East, and I would like to present SimDash, a Python simulation dashboard. So SimDash came out of the Social Sim project a little bit because we wanted to be able to visualize simulations that change with time. And so they worked by, one of the more common ones, one of the more common visualization techniques that is known as TensorBoard, which is one of like it's a type of simulation board, and we wanted to rival it with SimDash because TensorBoard cannot visualize simulations that happen over time, that change with time. So say that you had like a graph of a map or and you wanted to see populations, and you wanted to see how they changed from the 1970s, the 1980s, the 1990s to now. You could drag a finger along the along the bottom part and see how it develops. And so one of the interesting things that we wanted to have is a intuitive web UI that you could just run straight from a command line command, and it would generate a local host, and you could see the visualizations that you wanted to create. And finally, it's a free and open source Python package, so we wanted to have well-documented code, and it needed to be easy to read so that others could go in and change it themselves, and it's free, so it's easy for them to obtain and get. The features, I wanted to implement several features into SimDash, but I haven't had the chance to implement all of them yet. The first one is a fully functional database for storing all of the data and relative information such as the columns and the data types. Then we wanted to have a flexible data model for the visualization, so we could visualize scalars, maps, vectors, and how they change over time. So as like you see the visualization change as time changes. And then we wrote a couple input pipelines that help you see that, like a couple trial input pipelines like get PID, which will allow you to look into the PIDs that are being run by a certain user and see what they are running and how much CPU and memory usage they're using up. And that's just an example of an input pipeline. You could write your own for your own simulations and see the information that you would like to see. Then we wanted to implement once you put your data into the database, we want to implement a web UI that works with plotting the actual charts that you wanted to visualize, and it works with the command line commands. So you just say sim-serve, and it'll plot all of the charts that you wanted to visualize. Here's an example of the interface for what sim- is going to look like. On the left, you can see a chart that shows memory usage and CPU usage over a period of time, and you can see how a simulation has maybe used up a certain percentage of the CPU or something. And then on the right is one of the more complex visualizations, which is a map, and it shows data points, and then you can drag along the bottom and see how those data points change over time, over location, or over in their size. And here's a diagram of the software. So it starts with a researcher who either writes their own data input pipeline or uses one of the 7 ones that we provide, or they can just input the data correctly into themselves into the database. And then they write a configuration file which specifies which charts to make, as well as which kind of graphs and the encodings of the X and Y variables. And it takes the data and places it into the visualization interface. And then that uses Altair, which is a Python visualization program, and creates Vega or Vega Lite graphics on the web server, which is a local host run on Flask. And yeah, that concludes my presentation.
So my name is Zane, and my research was in analyzing protein sequence similarity networks. So I'll tell you what those are in a sec. Uniprot is a protein sequence database where researchers can upload any protein sequences they discover in their research. So what the team did before I came was they compared all the proteins to one another, and they built this table where we have protein 1, protein 2, and then the bit score is how similar they are. We only include them in this table if they're over a certain threshold. And then what I did is I took this table and I represented it as a graph or as a network where each node is one protein, and then the edges and the edge weights between those nodes are the bit score and how similar they are. So this graph actually had many different components. There's about 6 or 7 that were over 100,000 nodes, and there were probably about 1 million that were below 1,000 nodes. So we were looking at the bigger ones because there's more patterns that we could recognize, and there's less randomness. So the goal was to understand the structure of these similarity networks. So that means the connectivity properties, the structure of the similarity, so where are the very similar nodes and where are the less similar nodes within the structure, and then compare that to known network models. So we could see if there was any known network models that we could compare our data to. So examining the structure of the edge weights, so what we did was we built a graph called G alpha, where we made a threshold for what weights were going to be included in this graph, based on a proportion of the maximum weight in that component. So as we change alpha and we increase, you can see the number of components increases because we lose a lot of the connections, so the parts break apart. The average degree of nodes goes down because we lose a lot of the connections between the two nodes. And you can see these are 2 different components, but they have somewhat similar structures as we look at them here. So the next approach was analyzing more network properties. So the 2 important ones here are the degree distribution, which is just a frequency histogram of the counts of each degree. and then a k-core distribution, where a k-core is the largest subgraph of a graph, where every node has at least k edges coming off of it. So when we look at these histograms, we can see these, again, are two different components, but they have some similarities between the structures. So we can see the degree distributions sort of come down across like this, and then they sort of fall off at the very end. And in the k-core distribution, there's a lot of flat sections followed by very steep drop-offs. And even though they're not exactly the same, they still have those same features. So the third approach was looking at the k-core. We wanted to collapse the k-core into a single node, so we could see how this densely connected inner core of the graph interacts with all the nodes around it and all the edges outside. So what we did is we chose the first drop-off point where, so k equals about 100 here, and there's a drop-off. And we condensed that 100 core into a single node. And so here you can see before and after what happened. So after, it removes a lot of the drop-off at the end because a lot of the high-degree nodes have been removed, and it's a lot more linear without the huge drop-off at the end. So what are these results? Well, we found that they didn't fit any standard node network models initially, but after we removed the k-core, you can see this orange part is with the k-core removed. It's much more linear, and we can basically break it up into 3 different sections. And one of the things is, it might even be technically 2 sections, but because we chose 100 core as the cutoff, we sort of have some residuals left in this graph that's caused by choosing 100 core, because it wasn't exactly a sharp cutoff, it was sort of rounded off at the top. So what does this suggest about the graph? Well, it means there's some sort of dense core in the middle, and then a less dense core around it, and then some nodes extending off of the outer core. And if we go look at these k-core distributions again, they suggest the same thing, because there's sort of a large port where a lot of the nodes are included, and then all at once, a large portion of the nodes get dropped off. So that suggests that there might be multiple layers of cores as we get down to the highest k-cores on the inside. So what's cool about this project is the initial research is in biology, but I don't understand a lot of the biology that they're using. But I was able to analyze these graphs, and hopefully my analysis and results will help the biologists understand something about these protein similarity networks.
Hello, I am Matthew, and I'm glad to talk to you all today about pattern detection in high-dimensional data. So, over the summer, what we've been interested in is comparing similar populations that may or may not be dependent on, that may or may not have a diagnosis of opioid dependence. However, the greatest challenge we've been having this summer is getting access to the data in the first place to do analysis on. So for today, I will discuss the methodology that we plan on applying. So high-dimensional data is characterized by having many features, and these days, this kind of data is sort of collected without any sort of hypothesis to test. So it's a challenge for us to best make sense of this data. And what we're interested in is using an approach from topological data analysis, and we'll be using the map. We started by using the mapper algorithm. So what we want to do is go from our high-dimensional data to a weighted directed graph. So essentially, we're simplifying what we're looking at. And then with this graph that's easier to visualize, we can utilize optimization algorithms to discover sort of the most interesting activity, whether it's paths or branching activity. And then from there, we can look at what's the most important in our graph, and we can use that. We can analyze that. And what we want to do is find diverging subpopulation level behavior, and ultimately we want to be extracting hypotheses from this process for further predictive testing. And applications right now include plant phenomics and hospital patient trajectories. So I guess just to set the stage,
you can imagine that the graph we want to get as a process, I guess like a problem, an application problem we're interested in, would be sort of following events of, say, hospital patients over time. So let me first discuss the mapper algorithm that we'll be using. So we'll start by having a bunch of filter variables. These are variables that we'll pick out from our feature space, and those will form the high dimensional space that we'll be working with, and then we'll also be picking an output variable from this space, and I'll elaborate on that in a second. So with our space of filter variables, what we'll be doing is with some set size, meaning set like intervals for each dimension, and overlap between each interval, we'll be partitioning this space into hyperrectangles, and in each hyperrectangle with overlap, we will be using a clustering algorithm of our choice. And so this output variable is what's used for the clustering. And again, it's something that we pick based on our application of interest. And ultimately, after the clustering, we will have a topological object that easily translates to a graph. Essentially, vertices will represent cluster centers, and edges will represent intersections between these clusters, meaning that some point matches to multiple different clusters. That'll form the edge. So once we have this graph, we can set edge weights by the following. Find averages of this output variable in each vertex, and the graph will be directed from low to high based on the difference between these values. And here is sort of a picture just briefly showing the process. Imagine this is like higher dimensional space, the space of our filter variables. And then we get a graph like this. Imagine there's arrows, meaning it's directed. So we'll have this chosen filter. For applications, say it's time. So if you go left to right, we'll be maybe following days since someone was admitted into a hospital. And ultimately with applications like this, we might expect that we start from the stem, say like the whole population, like day 0 or something, and then we'll have these branching activities where subpopulations break off with their own behaviors. So yeah, this is what's called a flare, essentially the branching process. And so at this point with our graph, we're interested in finding what paths and flares are the most important to look at. So this just encapsulates the interesting flare algorithm. Essentially, we'll be having this backtracking process of accumulating edge weights, reaching branching nodes, and then dividing the score amongst stems. And this is something we sort of follow back to a branching node of our interest, and then that'll be a score for such a branch. And then for interesting paths, let's refer back to here when we talked about how, I guess for each cluster we picked the average and sort of took a difference, to sort of get the directions. So I guess we could do a similar thing with filter variables. Take the average and take the difference between them. Make it 1 if it's positive going this way or that way, 0 otherwise. And so what we define as an interesting path would be a path where if you follow on the directed edges, for every filter variable, you'll be monotonically increasing or decreasing. So that makes it interesting because we'll be noticing for all of our filter filter variables that we're sort of getting away from the average. So that's what makes it interesting. And then we have this interesting path equation, which applies for interesting paths and is non-linearly based off of the edge weights, again, which relates to the output variable. And so that constitutes an interesting path. So how can we find the most interesting paths in our graph? So there are different optimization algorithms. One example is MaxIP, which gets us the largest IFP value for some path. And so that gets us the most interesting path in our graph. And another optimization algorithm we can use is IP, which sort of partitions most or all of the graph into a set of paths, such that if you add them up, it maximizes the sum of their IFP values. So ultimately through this, we get the most interesting paths and branches. And from there, we can do analysis. So say, we have a stem and we have branching activity, etc. Say now we know the most interesting paths or interesting branches. What we can do is sort of do analysis by focusing on these vertices. For example, So I guess like with filter variables and output variable, we pick continuous valued variables. But say now if we're doing our own analysis on this graph, we can say make each vertex a pie chart or whatever we want, or like different, pick whatever outputs we're interested in, and we can do data analysis and try to learn information from this graph. And that'll ultimately help us to work on further predictive models. And that's it.
My name is Josh Priest, and I worked with Ravi and Madhav on solving decision problems for synchronous dynamical systems using Boolean satisfiability. So your first question probably is, what is a synchronous dynamical system? At its base, a synchronous dynamical system, which I'll refer to as a SciDS, is an undirected graph with each node having a Boolean state of 0 or 1 assigned to it. Each node has a local function that determines how the state will change in the next time iteration based on its inputs. So every single node will take in its current inputs at one time, and then all will change synchronously. So these SciDSs have some interesting problems that generate out of them. One of them is a fixed point problem. What this problem asks is if there is a configuration that is its own successor. So if this is the case, that configuration is a stable state of the SciDS. It won't change out of that state. Another problem is the configuration sequence completion problem. So given a trajectory for the SciDS with some unknown values, what this question asks is there a way that we can assign Boolean states to the unknown values such that it represents an actual trajectory of the SciDS? So these questions can be answered by exhaustively calculating every single configuration that could exist for a SciDS, but that's extremely computationally expensive, and it will take a long time, and it isn't really feasible on large graphs. So what we can do instead is all of these problems can be converted into what we call a Boolean satisfiability problem, otherwise known as SAT. So the nice thing about SAT is will return answers to these problems of whether they're satisfiable or not. So it'll return whether there is an answer to the problem. And there exists lots of open source software out there that solve these problems very quickly. So there's a few popular open source SAT solvers that can take in thousands, millions of clauses for these problems and produce solutions very quickly. So I wrote a Python program that takes in a synchronous dynamical system and a problem, converts it to a SAT problem, and then feeds that to a SAT solver and gets a solution. So these are some of the results for the fixed point problems. You would expect that certain functions would take longer than others, and we didn't really see that in our results. A lot of these look to be exponential according to the number of nodes, but it's hard to say because we don't have enough data. So I don't want to make any assumptions about the actual function of both the time it takes to convert and or solve these problems, or how big the actual Boolean satisfiability problems generated are. The CSC problems had similar results, but so SAT solvers, when they're given an unsatisfiable formula, will return the result very quickly. They'll decide that the problem does not have a solution and will return that in less than a second. So we have data for about nodes of 1,000, or graphs of 1,000 nodes, but we've recently been able to kind of extend that out, and these problems might take a while to run, but we can get more data sets. So part of what we want to do in the future is expand these experiments up to significantly higher numbers of nodes and edge densities, such that we can kind of generate more data and see some patterns in how long these take. We want to discover some trends of how different SciDSs and SciDS problems can affect these SAT problems that are generated, and how long they'll take to solve. And then at the end of this, we want to provide this program that converts SciDS problems to SATs as a free and open source program to the community.
Hello, I'm Joseph Alton, and this summer I worked on protein sequence annotation using convolutional neural networks. Okay, so just some quick background. I'm part of a team of Drs. Andrew Warren and Steve Eubank that is contracted out by FundyCat. Like, they contracted out like a handful of teams for this. Contracted out by ARPA, which is the information version of DARPA, essentially, on a project called FundyCat, which stands for the Functional Genomic and Computational Assessment of Threat, which is basically a project that's trying to translate DNA sequences to a potential threat level for a variety of applications. And so one of the ways to do this is by annotating the proteins with essentially what are called domains, which are functional units of these proteins. PFAM is a database that characterizes over 17,000 of these domains, and is kind of what we were using to kind of be like a benchmark data set for this. And the state of the art for this kind of annotation process is called InterProScan, which takes in all these thousands of hidden markup models, as well as consorts some other tools and databases to make predictions on annotation. And so we're trying to use convolutional neural networks to see how that changes prediction and increase efficiency and potentially accuracy of that process. So some background on the data. So we're working with amino acid protein sequences, and they come in kind of two forms that are relevant to the project. The first one is core sequences, which are essentially the domain sequence with very little noise flanking either end, and profile sequences, which have a high amount of noise on either end of the domain and between domains if there are multiple domains in like a query sequence. And so Hammer is a tool that we were using to emit training data essentially from the hidden markup models from the PFAM database. Its kind of architecture is shown at the top and its logo is shown at the bottom. Essentially he uses these hidden markup models and emits example amino acid sequences for those domains. So then my first order of business for the summer was just develop a wrapper to automate this training data generation. And so I'm just putting in the name of the domain that I want to get out, and it'll give me any number of examples in the core profile setting. Okay, and so some previous work that's been done in this project, some previous undergraduates as well as a current postdoc, Dr. Jacob Porter, we're working on this convolutional neural network. The architecture that they came up with is on the right-hand side. And it shows pretty good core performance, which is like predicting and annotating on core sequences. However, it showed pretty or poor performance on profile sequences, and that's a problem we're trying to deal with. As well as it can't hold all the domains without suffering some accuracy and F1 score. So we're trying to figure out how to deal with that. And so one of the first things they did for the summer was look at a recent Google research paper, which published on this topic. They had one convolutional neural network that basically was predicting on all these domains. However, we quickly figured out that it also had the same problem with not being trained on and predicting on profile sequences. And so that necessitated a model that would be able to only require the CNNs to predict on core or core-like sequences, was able to consult a number of CNNs because we can't put all the domains in one, as well as be able to annotate multi-domain sequences. And so that's where my work came in, the primary amount of my work. And so the solution for my project was to basically build a sliding window wrapper for CNNs with similar length domains. And so the process for this tool is shown on the top right here. And so we sorted all the domains by length and then took 100 length bins, it's just our first iteration, 100 length bins of these domains and trained a CNN on all of these bins. So we have like a set of CNNs to cover all of the 17,000 domains that we're working with. And so basically we take a query sequence and we will break it up into subsequences that are appropriate for the domain length for each convolutional neural network. And that makes it so that it's only predicting on core-like sequences. And so after breaking up and making those CNNs predict on all those subsequences, we get an array of most likely domains from each subsequence from each convolutional neural network, and use a greedy algorithm to take the highest probability non-overlapping domains from that output. And that's essentially the annotation that we are trying to generate. So the bottom left-hand side of this slide shows the histogram of the PFAM domain length. You can see there's pretty high kurtosis. So that shows that most of the domain bins are going to have similar length domains, except for there's some skewness on the right. So that might show some problems for the accuracy of like the bigger length domain convolutional neural networks. We have to figure out how to work around that. And then the heat map on the right-hand side, just a visualization of the confidence for each model for each subsequence. And so the models have different lengths that are associated with the domain, which is why there's a bigger like white region. Essentially, that's the section that it's not predicting on anymore because there's not enough left for the sequence to make a subsequence. And the greedy algorithm just takes the darkest one of these sections and then just nullifies anything that's overlapping with, and then continues that until it has a complete annotation. And so current results show that even though all these CNNs have a pretty high F1 score and accuracy on the domains that they were trained on, there is a high rate of false positives within this wrapper. And we are seeing that a lot of that is being caused because these CNNs are overly confident about predicting that one of their responsible domains is in a subsequence that is not contained in. And so we're trying to figure out why this is happening, and some potential causes are that it may, well, basically some solutions that we're trying to show are that adding an adjunct class, basically, so that if we have these CNNs, we can also train them on some subset of examples from all of the other domains, so that each CNN can kind of decide whether or not a subsequence is something that it should be responsible for, as well as we're seeing some of this, some of this high false positive is caused by similar length CNNs trying to predict on domains from each other's responsibility. And so we're going to try to do some overlapping in the responsibility, basically which domains each CNN is responsible for, to try to mitigate that cause. So other than that, we're also going to look into improving efficiency using GPU and multiprocessing, as well as improving the interface as a tool for future work. And that's what I'll be working on in the next couple months. So that was my project. Thanks.
Hello, I'm Andrew, and I was working on doing time series analysis on influenza data, specifically supporting the CDC Flu Site challenge. And so the CDC Flu Site challenge, they've been doing it since 2013, and they're asking teams of researchers to forecast the influenza rate, and actually several targets. So there's the influenza rate, the start of the flu season, which is determined by a baseline that's established by the CDC, the peak intensity of the flu season, which is just like the highest percentage it'll reach that year, and the week in which it'll reach that peak intensity. And so looking at the mechanics of the actual challenge, this is the data that the CDC publishes weekly on the flu rate. Then they they publish data nationally and by 10 regions established by the Department of Health and Human Services and by state. And so they update this weekly, and this was the primary data set that I used to incorporate and like train our time series analysis models. So for the implementation of my specific product, I created a harness that can just kind of train and like test, like not just one specific univariate model, but like just a wide range of univariate time series analysis models, because these are pretty simple models, and so it helps to kind of look all over the time periods and like see like, okay, how would this model perform for the 2017 and 18 season, or how would this model have performed for the 2014 and 15 flu season? And so, the CSV file is an example of an output, and that would be the score submission that you submit to the CDC challenge. And the way they do the scoring is in bins. So your model could return a point prediction, but you could have a level of confidence in your specific prediction, and you'd say, oh, like I'm extremely confident that the flu rate will be 2.4% or something. But then you would for my models, I wrapped a normal distribution around it so that there's uncertainty in your prediction. And there's a few steps to making your model. So you got to collect the data, then pre-process, train a model, and then output, generate plots, or do other error analysis. And so my model, or my harness, will handle all of that, and then the actual model that you can kind of plug in will run in this command line script that I created. And so that's just a sample usage of the command line script using a simple autoregressive model, and it generates a prediction for the 40th epi week of the 2018 year using training data from 2002 until 2018. And this is a sample plot generated of the same thing. This is a 1 week ahead prediction using a simple autoregressive model on the 2018 flu season. And so, I mean, you can see that it tracks decently, especially along the beginning and ending periods. I mean, obviously, the harder part to actually predict would be the middle part, where the fluctuations are greater and it can go down. But like when you're looking at the end and the beginning and stuff, it's not, like obviously it'll track pretty well. And so the advantage of having like a harness is that the flu seasons of like 2009 and 2010 were just particularly intense. And so if you wanted to, you could immediately backtest any univariate model that you come up with on those crazier flu seasons and get results for that. And so, I guess in terms of future implementations, we would want to look at transitioning from univariate to multivariate models, because the primary data set that we're using right now is the flu data, which is published weekly. But if we want to incorporate multivariate models, we could look at the correlations between HHS regions and how they would impact like a national forecast. Or like if you look at geographic correlations between HHS regions, you could arrive at a better prediction for a specific HHS region. But when you start to consider those multivariate, when you start to increase the order of complexity of your models, it becomes obviously a lot harder to create like a clean scaffold for it that you can just immediately like click any option and like create output. So that would definitely be an order of complexity that we'd have to look into. And then as far as challenges I faced trying to implement this harness, where one of the challenges was data validation, because when we were using the primary CSV file, like not all the data is just there. Like data is missing. Data can be updated because of backfill. So like hospitals will regularly, like they'll publish a report, but then they'll send in additional data the following week, and then okay, now your model is has changed. So as the harness stands right now, it's not very robust, and that like small, like a typo or something could just break the harness and you just return an error. So we would want to improve data validation in that regard. And then date conversions. So the CDC uses epiweeks, which is defined as the first epiweek of the year starts on or ends on the first Saturday of January, as long as it falls at least 4 days into the month. So essentially the first epiweek of the year could start not on January 1st, but like December 29th or something. So you have to account for that, and when you're trying to convert between those dates and like the dates that like in pandas when they recognize those dates. So that's definitely something that I had to take into consideration. And then ideas for like future implementations and future iterations of this. We definitely want to work on increasing the data validation and also like adding more error analysis of like these models and more more ways to just quickly analyze and like generate analysis for our predictions. And that's my project.
Thank you. Thanks for your summary. And thanks to all the Institute people that participated in this as well. One of the things that everybody uniformly has been impressed with actually is you. You've done a wonderful job. You've made an impression. We are new. We're just getting our feet on the ground, so we don't exactly have the building filled up yet and a variety of things like that. But I would like to welcome all of you to remain and continue to be a part of us this going forward. There's a lot of different things that we do. As I say, sometimes we look like the Department of Everything department because so many different topics get worked on. But actually what we're working on are the class of problems that you really can't study unassisted cognitively. And I'll give you examples of this as we go forward, but my favorite current example is it doesn't have anything to do with biology, but there was a testimony of a person from a lending company, one of these short-term lending companies, was testifying before Congress, and he was being sort of harangued by this congresswoman, and she, in her other life, was a professor, I think, in an economics or a law department, and what she studied was payday lending. That's what she studied from an economic and legal point of view. And so she went through this long list of criteria that they had that constituted fair warning to these people, because the argument was as well, I mean, we gave you fair warning. I mean, the fact that you get into a 200% loan is like your own problem, right? I mean, we told you. That was sort of the argument. So she gave him how you actually present what the terms of the loan are, and she said, what's the APR? And so the guy incredibly condescendingly explained to her that I guess he didn't know who she was, didn't know that she'd actually written textbooks on this topic. But he went through, well, it's a technical matter and it's kind of hard. But she said, okay, well, can you tell me what it is? And he said, well, in this situation, this hypothetical situation, I don't know what it is. She said, oh, I took this off of your current loan. This is yours. I just want to know if you know what the APR is. It's hard to calculate. And so she gave him a calculator. She had an aide give him a calculator, and he couldn't do it. And the point is, there are really seemingly everyday problems that you really can't do unassisted, at least not in a short period of time. And some of them have to do even with doing important things for the good of us, which I just want to point out is part of what the new
I don't think it's a new direction, but it's the explicit direction of our president. I think it's actually not a bad thing to bring up, is that we really ought to think about the value of what we're doing. And so what I'm saying is that we have incredibly important problems in front of us, much more important actually than this one, that we can't do unaided. And not to put too fine a point on it, I don't think we know what we're doing when we're producing the platforms that we use to think about them. And building machine learning widgets that are super cool. And teaching things to recognize faces and understand what you're saying and stuff. All that's really interesting, but it doesn't cut to the problem of how does this help us reason and think about the problems that we're confronting that are too important to write off and too hard to think of unassisted. We need this cognitive microscope in some cases to really get inside these systems and understand how all these pieces are producing something, and we need, I guess, a telescope as well to sort of look at the system as a whole. And it's basically that problem that I want to talk about. All the topics that you guys worked on this summer are exactly those kinds of problems. They're problems that without this platform you can't think about them. With the platform, supposing that you were ever endowed with the authority to do something, I challenge you to say, what exactly would you use this information for? It's not so straightforward, really. You have to be careful. And it isn't even you. I mean, the newest systems, the new system concepts are that these things are distributed among us to induce us collectively to do stuff. Even if it's just buy more shoes from that guy instead of that guy. And we don't know what we're doing, which is bad news in a way, but it's really great news for a research institute. And it's great news for you because the reason that this is happening, a major part of the reason that it's happening, is that the level of connectivity in these massively interacting systems is happening through the ICT layer. There's nothing virtual about this layer. These are very real interactions. They're as much a part of you as your peripheral nervous system is, but they extend your agency, they extend your mind, and we have to figure out how to engineer them. So they learn, they improve, presumably we're a part of them, and so it's worth considering the fact that these systems are studying themselves. And we don't know what that means either. So, there's plenty of opportunities around the world to look at things. I will point out that, and I don't say this is not, there's nothing political in what I'm saying here. Nothing. I want you to repeat that to yourself before I say it. Nothing. This is a technological statement. Huawei has a thing called the Safe Cities program. It's a customer.
So somebody, can people mute their microphones?
You might have the power there to mute them.
I don't want to touch it.
Sorry.
Before it became worse for them.
They got it just in time before we could see who it was.
Victory at last. So what does this have to do with what I'm? Let me just go on. All right. So the name of our institute is Biocomplexity Institute, and I'll just say that the way I see it here, and I think generally the way we are here, is an information and computational account of living systems, from adaptive molecular encoding, through social interactions among organisms. And so the example that I use, and I think I may have actually said it to you when you arrived, so if you're hearing it twice, I apologize. But if you look at antimicrobial substance and policy associated with antimicrobial substances, you see that things like laws and social norms how we allow manufacturers to make things, how they can sell them, who buys them, why they buy them, whether it's detergents or over-the-counter antibiotics or prescription antibiotics. All this complicated mess of stuff winds up with affecting your immune system and the microbial environment that you're in. It affects livestock. It affects water supplies and soils. It puts bacteria in continuous contact with low efficacy antimicrobial assaults, and eventually they change. And they become intractable. And so you see a relationship between social systems at the level of laws, behaviors, activities, choices, and it extends down to the adaptive properties at the molecular level, at the genomic level, on the microbes themselves, and probably us as well. That scope is normally not viewed as a problem. There's lots of legit problems sort of along the way, but the whole thing, so what I'm saying is that kind of problem is a legitimate problem. And that's what we study here, that kind of problem. Large, integrative, multi-scale problems are legitimate problems, and they do have form. For example, and I'm going to go into this today, I took all this out of this talk, is that there are abstractions as dynamical networks that are extremely important in all of these. The abstractions have to be applied, but they're abstractions. You know, and so there's a big theoretical program, there's a big computational program, there's a big modeling, as well as all these vertical knowledge levels that we need to, knowledge areas that we need to change our level of capability in. And that really does form the basis for this kind of science. We couldn't have done this without the advances in computation that we have, and there's a coevolution between the mathematical and theoretical principles, the computational principles, and the products and environments that we find ourselves in. So that's a massively interacting system because every one of these pieces is interacting in lots of different ways. And all I'll say is that brains, networks of brains, extended brains, augmented intelligence, whatever we were just talking about, those are also massively interacting systems that live in this scope. So science, historically, at least since people like Newton, is in an interesting scope where they love it. We love explanation from elementary relations. But living systems resist this program. When we try to reduce life to reductionist programs associated with molecular science, I mean, we're confronted with Enrico Fermi's comment that says, well, if all these are so inevitable, where is everybody when he looks out at the world? That's why Enrico Fermi was a really smart guy. It's like little things like that. It's like, sort of leaves you speechless. Because, you know, one possible answer is we don't know what we're looking at when we're talking about life, but with respect to, you know, maybe there's other things. With respect to mental phenomena, how do kinetic, thermal, field objects spontaneously become cognate is, I mean, here we are. Like, we don't know that either. So deciding to design cognitive systems is kind of interesting. Since we don't know how a bucket of swamp water starts thinking, it's pretty important to know because we're all just a bucket of swamp water. If you want to take the reductionist perspective. So, if it's so inevitable that we do begin thinking, and apparently it's kind of inevitable, so how do we recognize it? How do we know what we're looking at? Well, how to know what you're looking at isn't so easy. There's examples. This is just one. I mean, there's so many examples. I just picked this one because I had a slide that made a long time ago. So there is a species. It's a African grasshopper that when they, you know, there's at the beginning of the genomic period, everybody just was, to some extent this is still going on, but it was really like sort of what was happening. Everybody just running around grinding everything up and sequencing it. I mean, it just was crazy for years. And so, anyway, they were grinding up grasshoppers and sequencing them. And they noticed that they had these 2 samples that were supposedly different things, and they got the same genome. So they thought they'd mixed up the samples, and so they went and sampled again. And they did it several times. They realized that they were the same genomes. And if you take this little guy and make too many of them, or if you stress them like this, when they're in the larval stage, they turn into that. Now those are very interesting creatures because they reproduce this way. And they don't have the same exoskeleton, they don't have the same leg geometries, they don't have the same mandibles, they don't have the same protein structures, and they don't have the same keratinaceous exoskeleton. But they have the same genome. And they're an adaptation, an external adaptation. So where is the information, and where's the information in this? We're not talking about we're not exactly talking about evolution. We're talking some sort of like very large riboswitch thing going on here, right? If you don't know what a riboswitch is, it's at the RNA level where you have very similar things to that. You change the environment, all of a sudden the same sequence makes a completely different functional RNA. And so the question is, where's the intelligence in this system? Is it coded on the DNA? Is it between the DNA and the code? Is it on board? It's not a simple question. So when we interact with things, we interact simultaneously. If we're massively interacting, we interact simultaneously a lot of different ways. For example, at one level, we interact kinetically. So those are, I'll call those simple interactions. Billiard balls do that. One of the most important, the reason I bring it up is that billiard balls can't lie. If you just think about it, this comes up in a minute, so bear with me. Billiard balls and other kinetic interactions cannot deceive each other. There's no such thing as a deceptive billiard ball to the billiard ball. It might be deceptive to you, but not to the billiard ball. Okay? But there's mediated interactions as well. So the interaction where I tap my knee and my leg, that's a reflexive interaction. And then there's deliberative interactions where I take information about the world, I form a state estimate, I continue to compare it against the world, and I base my actions not on the data, but on my state estimate associated with the data. So it's mediated by my state estimate. So if I'm playing poker, or if I'm flying a fighter aircraft, or if I'm buying and selling stock, there's an aspect of what I'm doing that has to do with the current price, the current configuration of things on my radar screen, or whatever the situation was. But then I have to say what it means and what those things are, and that's what feeds sort of the left-hand side of the rules that drive my behavior. That's state estimate. And in that situation, I can deceive you. In that situation, you can have the locust or the grasshopper as your mediating state, and you won't do the same thing with respect to it. So you can have, so when you have mediated interactions, what you have is several things, but one of them is that they're non-unique. Now, and there's another problem with them, and then this comes up at the end of this, and I'll bring it up now. It has to do with levels. You can always play this game. I can write an agent, like in a software, I can write a simulated agent that's an electron. And it can go up to some atom, and it can ask to have the atom tell it what its electron configuration is, and it can calculate things and judge whether or not it wants to attach to this thing. You can do all that. Now that isn't what electrons do, but I can make it exactly mimic, Hund's law. So there's a simplicity criterion that attempts to be put into place. There's a guy named Morgan that made this really, really clear. You never appeal to a cognitive state when you can get away with not appealing to a cognitive state. in an explanation. The problem is much harder, though, than Morgan apparently grasped when he made this injunction. Simplicity criteria work really well for billiard balls and Lagrange paths. They don't work very well for cognitive systems because cognitive systems exploit the non-uniqueness and they deceive one another. I mean mediated systems. Well, cognitive systems too, but mediated systems. And this doesn't start with humans. It doesn't start even with camouflage on frogs or something. It starts with viruses. Because viruses deceive cells that they dock on. They're constructed to perform a deception. So as soon as you have autocatalytic systems, apparently they start lying to each other. The only way that works is if you have non-unique representations for a given piece of data. And enough evidence to take the wrong one. The point is, and what it has to do with what we were talking about, is when you're building the tools like you were working on this summer, and that's our part of our program is thinking about these as tools. They can be misused, and they can also be misled. They can also do great good. But if we're going to engineer them, we have to take this into consideration. And I think that one of the most important problems right now in the world has to do with the use of computational technology underlying situation assessment and decision making. It's evolving and it's important and it's worth thinking about. And it is one of these questions that it's a legit question in its large form. And it will affect you in a big, big way for the rest of your lives. So, if deception exists, non-uniqueness is being exploited, and the system must involve mediation. So one test on whether or not you have a mediated system isn't a simplicity criterion, it's whether deception occurs. If deception occurs, you for sure have a mediated system. It doesn't mean it's having great thoughts in writing Shakespeare and things, but it does mean it's mediated. So, line is an indicator. Okay, so general cognitive process architecture. So, there's a functionalist assertion here. This is to the theorists of us. Any mashup of anything that creates this: sense, interpret, make a situation assessment, form decisions, assign actions, and take an effect. Anything that can do this is cognitive. Anything. So the question, like, what do we look for? We look for that. Another assertion is this is how it's related to the concept of agency, whether you're a corporation or a person. Things that do that, they possess agency. They can interpret, interact, and decide. They possess agency. Okay, so does such a thing have to reside in, say, a person? It doesn't have to be a person. Any particularly doesn't reside anything. Can we extend it? Well, corporations are agents, and they're composed of individuals under limited liability, constructions in the law and stuff with respect to. So apparently, no, you don't have to. It doesn't have to reside inside an agent. Well, okay. So Otto. This is a philosophical foil of Andy Clark I'm blanking on the other guy's name. Chalmers, yeah, David Chalmers. In extended mind. And so the extended mind theory basically says, so Otto has a problem, dementia, or anyway, his memory is flawed, and he wants to go to the bakery. Well, he doesn't know he wants to go to the bakery, but let's suppose he wanted to go to the bakery. the problem is to go to the bakery, you have to remember where the bakery is. You have to remember 2 minutes from now that you wanted to go to the bakery in the first place. You have to know how to get there, and so on, right? But there's something wrong with Otto's mind. So every couple seconds, every couple minutes, I should say, this thing honks, and you look at it and goes, Otto, you have a memory problem. You're going to the bakery. You're going to be on this street, continue down this street. You know, you don't have to explain to him what a bakery is. He remembers that. He doesn't know from every couple minutes, he forgets he's going to the bakery. So the argument essentially is that if Otto could do this before he had his memory problem, and you could, in some sort of architecture like the one we just drew, write down what Otto had to do to, go to the bakery, had to remember the bakery, make a plan, decide where he was, look at his environment, figure out if he's on the route, change his route if he wasn't, go, do all that. If it was cognitive before, when you replace something with this memory thing which isn't in Otto, it's still cognitive. It's an assertion that's as simple as that. So that would mean it doesn't have to reside inside a person. That means all the tools we're making, all the things that you're engaged with, all the things that tie you together are of this kind, if they're making you able to think about things you weren't able to think about before effectively. It means in objective terms, if they increase your ability to do it and perform it, it isn't that they're intelligent, but this thing that, this cognition is made more effective, it is more intelligent by virtue of its use. But it's also not just you anymore, and the characteristics of your detailed interactions with the world have changed because you didn't have to used to know how to use this, read, any of that stuff. You just thought about going to the bakery. Now you do. So if Otto couldn't read, or Otto hadn't learned previously how to use one of these things, then Otto's in big trouble, right? Because it changed what he had to do. So this extension is much deeper than just saying Otto's extended. It's a new system completely. And just like your neurons don't know what they're thinking, you can be a part of systems in this way, and you don't know you're a part of the system. In fact, if you look at democracy, American voting democracy, which is in the Constitution in really a lot of detail, what it does is it distributes the sovereign's mind. And it builds into the Constitution, for example, the post office. That's a technological structure for a distributed thinking system, and the post office was a communication system for it in 1776. It's a distributed thinking system. It uses very specific methods, but that's what it is. So payday lending testimony, I already went that. Route planning. Okay, so now everybody has their GPS system, but instead of just pushing the button and accepting the route you're going to take, you accept that route, but you also post it on a cloud where everybody else's routing system can look at it. So now you can predict where everybody intends to go and when they expect to get there. So everybody reroutes. We don't do that, but we could do that. Lane changing, 2 automatic driving cars are there's an empty lane, they want to go. How do you decide who takes that lane is going to be something that involves both thinkers. So among people and things and teams and social networks, we're going to have networks or structures that look like that control architecture I told you about. So now you're going to say, let's take Otto's example and say, we're going to have a group of people that collect data, find out where bakeries are, find out where the best bakeries are, get maps, do all this kind of stuff. Somebody else is going to make a situation assessment and decide what the best route for Otto to take is. Somebody's going to say, these are the options, and he's going to select one and he's going to go on his route. And there's all these different people and things going on. And that system is intelligent in exactly the same way also. So now you have a team, and so if I make a platform that teams that perceive, make situation assessments, plan, interact, and do things, for example, like command operations centers in the military or something, those things could be represented and understood to be cognitive as well. So we have distributed cognition is possible. decentralized cognition, like swarm-like behavior is decentralized cognition, but you can have things like a GPS system that everybody gets their position from, and so they might be centralized even though they're distributed. So if you shut down the GPS system, then you would dysregulate the distributed system anyway. So distributed and decentralized, not the same thing. We're building these things, we're making these things think, we're putting them in very, critical environments, and I normally don't see things broken out this way when I read about distributed control and distributed cognition and the use of AI in emerging world, or the study of complex systems. I've already talked about riboswitches, multifunctional RNA. There's also fungi that attack things like spiders and ants, other insects. They take over their nervous system, their peripheral nervous system. They replace their peripheral nervous system. In the case of leafcutter ants, they cause the leafcutter ant to climb up onto a leaf, lock onto the leaf, and they eat the ant from the inside and spray down fungi spores on top of the other ants. They take over the ant's behavior. They take over the ant's nervous system. They make a zombie out of the ant. The fungi, individual fungi, do not know what they're doing, but they replace the nervous system of the ant. So they use the actuators of the ant. They replace its mind with a distributed mind. I'm just pointing this out because it's important to realize what we're talking about is, first of all, already happened in some respects. It's sort of the Enrico Fermi thing. Is like, well, what are we doing? We are expecting cognition to exist in some sort of compact form, like inside a software object or inside you, but it exists among us, and it's legit, and that's what cognition, and we're building computers that think, and it's in computational systems that think, and it's really important. Morphological computation is important. I'm just going to just say that, and I'm not going to go into an example because I'm sort of going too long. Man, I've already talked about this stuff. So engineering these systems is more than just saying I'm writing machine learning algorithms or making some adaptive componentry or doing facial recognition or something like that. It's really a lot different than that. It's extremely important to realize that these are pieces, but the cognition that is appearing is not in this algorithm or that algorithm, and whether it makes something more or less intelligent depends on a lot of things. They're important, and they will lead to the improvement of cognitive systems, but we need to be just as diligent about studying the cognitive systems from an engineering and science perspective as the widgets, because they will not just bottom up, spontaneously do this themselves. So we can wind up with very unencapsulated intelligences, ecological style systems that are cognitive. It sort of explains maybe some of Fermi's questions, as we're buried in these things and we don't even recognize what they are, but composed among us and others. Markets are examples perhaps of something that could have a computationally cognitive interpretation. And these. Okay, so bringing meaning to extended cognitive systems, meaning, what do I mean by that? Is that normally we think of you as a vertex in one of these sort of networks and all your properties associated with some mapping. Instead, what we're saying is these are all the different ways that you can interact, and what you are is essentially this line, this collection of pointers into the world. So you are inherently distributed. And you're connected to the world, and you're connected through the world to the representations of the world that you're connected to. So you're connected through the wireless communication network, and you're referenced by abstractions, maps, of the communication network that you can reference and use, and this thing just turns into this really interesting notion of a distributed self. There's no requirement that you are somehow consistent among these levels. So this is an example in philosophy and phenomenology. That was a mathematician named Rhoda pointed this out. I think it's a really interesting point. There's a concept in Husserl in phenomenology called Fundierung, and Fundierung says that you can't reduce logical items in the world to logical atoms, which is a problem for the philosophy of science, but because you have facticities in functions that can't be derived from one another. And so the examples he uses are people that are complicatedly in conflict with themselves. And so you can have somebody who's incredibly loyal to his country, his family, extremely diligent, hardworking, intelligent person, but he also happens to be, Goebbels. So there's a discontinuity between these kinds of things that are entirely possible. Where does meaning come from in the world? Sorry, I'm having problems with this. Is the meaning in the data? Okay, so rather than continue to watch this I'll let it run, but everything that you're thinking right now is coming from you. This doesn't mean anything. It's important to keep that in mind. And then I'll give you an example of what I mean by its sort of basic importance to the whole notion of what data is in the first place. If I just took one of those figures, like the little ball or the triangle, I left out every single thing else, and I showed it to you once. And I came back an hour from now, 2 days from now, a week from now, and a year from now, and I showed you the trajectory of the thing. What I asked you, I gave you like a choice. I could say, is it going to stay where it is, go forward, backward, left, or right? And I just asked you to do that. But you'd only seen the ball, like the little ball, say. And what I'm going to do is I'm going to calculate how often you're right. And you've seen it once. Then I get some number. Now I add all that other stuff to another subject, and I show them that. And then later, I show them the same ball and ask them the same questions. But now they have a story. And so the notion is, the hypothesis is, they would actually be better at predicting, they would be better at predicting what happened to the ball over time. Now why would that be interesting? It's interesting because you remembered more by adding data. Normally, memory viewed as a capacity. If I ask you to remember 2 things, you remember 2 things. It declines over time, but if I ask you to remember 12 things, you don't remember them very well and it goes away. If I ask you to remember 200 things, here I'm adding information and you're remembering more. And there's lots of versions of this experiment you could do, which I won't bore you with. But the point is that meaning has a quantitative effect a quantitative effect on the data. It's related to the interpretive action of the situation assessment that underlies the mediated behavior. And you can change its value by changing how you view it. So I can quantitatively change your data environment by messing around with your mediated state. What I'm saying is your mediation of that information has quantitative impact on the information value of the data, and it is not contained in the data itself. It's contained in the mediated state. And the manipulation, there's sort of nothing new in this in a way, but the manipulation of data, and outside the manipulation of data, that's the manipulation of cognitive framing for situations, is extremely, it's the foundation of propaganda, it's the foundation of many, many kinds of deception, and it underlies all of the creation of these advanced cognitive systems to the extent that they're mediated, deliberative systems that we're making. Do they scale? Well, we're building things that are pretty big, that interact, and that I would argue are generating layer after layer of legitimately cognitive things to the extent that we just do it. This is something that was done a while ago, but it has a lot of pieces that interact, and it can go into a lot of states, and if it was used to understand somebody with it's a biological model of the gut, and if you were using it, and it's at the individual cell level sort of thing. If you were using this for a person with the autoimmune disorder or something, it could get super complicated and you would be deceived. So we use the word intelligence, so it scales. So it's the tools we make have these properties, whether we realize it or not. They're important to consider, and we need to worry about it. Intelligence is competence. Cognition and intelligence are not the same thing. We talk about artificial intelligence. We really are talking about protocognitive things like memory, adaptation, something. We're talking about something that isn't intelligence per se, but if it was in a system, it would become more intelligent or something. But we can explicitly build cognition. And the question is whether it's performs better or not. To that extent, it's more intelligent. I would argue, not argue, I mean I would simply assert that the tools we're making actually do make the users more intelligent. We simply are more intelligent for their use. It doesn't mean we're better, it doesn't mean anything else, but it does mean that we are more intelligent. And I think, ironically, this leads to an environment where we understand ourselves even less, potentially. The need for the research about these platforms for the study of these massively interacting systems, to understand them in terms of the platform themselves, like not studying the epidemic, but studying how we know about epidemics is important. Critical even. And that's what we need to engineer toward. We need to engineer toward competence, not widgetry. I mean, we have to build the widgets, but the system level understanding of what their competence is critically important. It's not simply predictive modeling or something. It's much different than that. Okay, so I'm going to be done in 5 minutes. This is luckily the last slide. You'll be happy about that. Well, no, there's another slide. But okay, this is almost the last slide. So, does everybody know what Russell's paradox is? Does anybody know what Russell's paradox is? Give me a sense of who knows what Russell's paradox is. Okay, good. Okay, so then I'm going to explain Russell's paradox. But I'm going to do it in a way that is not restricted to Russell alone. So there was a great optimism at the end of the 19th century, at the beginning of the 20th century, toward the program of science in the 20th century, the reductionist program in science. There's huge optimism. for very good reasons. And if you think about everything that happened in the 19th century, it's pretty amazing. Steam engines and factories and industrialization and even telegraphy and all kinds of things, just astonishing advances. If you view medievalism and its transition through just scientific understanding, it's a pretty interesting time. So there was a lot of enthusiasm and also a lot of certainty. And so there was a program called logicism, and one of the premier voice of logicism was a mathematician, logician, philosopher named Frege. And he spent his life in pretty much disrepute in the mathematical community because logic isn't legit for mathematicians in the 19th century. Trying to ask the question, what is a number? Which is an odd question if you just view it like that. But he wrote two major sort of works. His first work was published, and then 20 years later, 25 years later, his second work was ready to be published. There's another giant in late 19th century science named David Hilbert. He gave a very famous talk on December 31st, 1899 about the program of science for the 20th century. It's everybody that studies, physics and stuff knows about Hilbert and knows about his talk and his problems. He cast it as a list of problems that would be solved in the 20th century. And the structure of this had to do with, first you would derive arithmetic from logic. And then you would get algebra from arithmetic, and you would get mathematics basically. Then you would get physics, and then you would get chemistry, and you get the rhythm of this, right? And it all started with Frege. It all started with the logicism of developing arithmetic from principles of logic. And he used set theory, which is pretty primitive. You need an element operator, you need intersection, you need negation. I don't think you need anything else. Union. I think you need union. I don't remember all of a sudden. Anyway, so Russell was a preeminent scientist, like Hilbert, more of a philosopher and a mathematician, but just a preeminent scientist. And he was a great proponent of this new program, and he was a great fan of Frege when many people weren't so inclined. And Frege sent him a copy of his book, his second volume, his life's work. I can't remember the name of it. And Russell read it, very enthusiastic, wrote him back and said, Golub, this is astonishing, and so on. So I just have one question. I just can't figure this out. Could you please help me with this? Suppose A is the set of all sets that do not contain themselves. Does A contain itself? Frege wound up in a mental hospital. This undid his life's work. You can't do what he wanted to do, apparently. Russell was working on the Encyclopedia of the Unification of Science, Volume 1, a guy named Whitehead. Volume 1 was the only volume ever written. They never got to the chemistry and one. But it also led, to the concept of logical type. Church's thesis, Turing, Chomsky, modern computer science, everything. For some reason, we still are intrigued with reductionism, but this is a big problem. So, with respect to mediated systems, if one mediated system is reading and controlling another mediated subsystem, we've agreed they might be doing that. If R regulates systems A that do not regulate themselves, does R regulate itself? With very simple definitions, is just a version of Russell's paradox. So, that's kind of interesting, and that's what it means. As we design intelligences, they'll be typed. This typing will result in authorities, differential authorities, notions of publicity, privacy. These will become technically managed and controlled matters of design and theoretical understanding of these things as cognitive systems. We are not going to get from a basket of widgets controlled, engineered, cognitive systems. We're going to design them and create them, or recognize what we have accidentally made and correct them. The question is, how will that be done? And what does it mean to this complex relationship from biocomplexity levels, from policy to molecules? And we need to understand this, and we need to understand it, and understand that it is legitimate science. It is a legitimate program of questions. And it is upon us. It's upon you, for sure. I'm kind of old, so there's only so far this is going to get before it doesn't really matter to me anymore. But I guarantee you that it's not going to slow down for you. Thank you.
Great talk. So we'll take questions. You have plenty of time for questions. So go ahead. Yeah. Please.
So can you give an example of a design intelligence? So you gave a number of examples, and they're generally evolved or sometimes accidentally designed systems. So what, maybe it doesn't exist yet, but what would it look like? What's an example of a design intelligence of the way you're describing?
That currently exists.
Okay.
That's what you're asking?
No. Or might exist.
Okay. Well, I can talk about one that currently exists that could get... Okay, so right now, it's not universal, but it's sort of common that if we're talking about having humans in contact with, say, AI, that we think of it as sort of like a human factors almost kind of a problem, right? It's like the interaction of humans and artificial intelligence. And there's lots of examples of that, right? But I would argue the right way to think about it is simply it's a new cognitive system. So an example of such a thing is semi-autonomous, something that I worked on 35 years ago, more than 30, never mind, really a long time ago, was fighter aircraft. That in an attempt to reduce the tempo and workload of the crew, but retain human authority over the release of a weapon, used artificial intelligence, but it just changed the whole airplane, right? I mean, it wasn't AI plus an aircrew. It was they didn't have the same tasks anymore. They didn't do the same thing anymore, but it was cognitive. I mean, it had a sensor environment. It supported situation assessment. Some of it was done on board the computer. Some of it was done, especially interpretive things, with the person. Final decision authority was reserved for the person, and many other things. So it was completely designed, but it wasn't viewed as cognitive. It was viewed as a human factors problem. And that works against you ever understanding the thing as a design system. Because you very naively start crossing wires that shouldn't be crossed and sticking things in because they're new, but they actually make the system stupider in an objective behavioral sort of way, and many other things. Automatic trading systems, high-speed trading systems, are embedded intelligent agents inside markets, which are arguably distributed systems that evaluate the value, relative value of things like securities. And it completely changes them. Automatic trading systems, poorly done, have created market crashes and all kinds of problems in the last few years, but we don't view them as agents that they are. We view them as problems in an algorithm, in this widget or that widget, and we don't think of it as a system that has failed. We think of it as an algorithm that somebody irresponsibly did wrong or something, right? So that's something that I would say is it's not exactly accidental, but it's important. And really important right now, I would say, our voting systems, which are part of this extended cognition that the Constitution in this country set up, are in danger, right? We know from Arrow that we don't have Kenneth Arrow, not from Cupid or something, but Kenneth Arrow. We know from Arrow that there's no such thing as a completely fair voting system, right? Or even a contradiction-free voting system. But we use voting systems as a rationation tool, a computational tool, in a distributed cognitive environment. If you manipulate how the data is interpreted or the data itself, you can manipulate the voting system. And I think it's pretty clear on all sides, all over the world right now, that there's I don't know if it's an instinctive recognition of this or what, but it's happening sort of everywhere as these systems and as people's everyday environment is more and more connected to the volume of information that comes out of this ICT layer. Right? So we're interacting in the ICT layer in a novel way. These things are exploiting that, and they're basically altering the survival value, or at least the rationale of a distributed cognitive system. You can agree with it sometimes and disagree with it sometimes, but the fact is it's changed. And it's best, I think, to understand it as a distributed platform. The last thing I'd like to say is that everything, when you have, like, if you have a command center, where in principle you could just have one, person that just has some incredible capability, and you just give them all this information and they just make decisions and everything works out. But the fact is that there's no way to do that. So you break down what that person would do into a staff, and they have roles and they have platform that they, somebody collects data, somebody does this, somebody does that. And you change the platform, you give them tools, and together, I would argue that those things are an extension, of whatever, the commander's intent or something, right? They're cognitive. So I don't think they're unusual. I just think that they're not understood, and they're haphazard. And as the world gets more connected, the consequences of that are worse and worse. And there's no reason to let that happen. And when we have things like molecular engineering on board individuals and a variety of other things, I think we have to understand this as a rationating environment, or we can't. Or it is us. We are one of these systems that has to, study itself, and we just don't know what we're doing. I think it's a really important thing to accept and to try and remedy.
So the message I got from the Hilbert piece and the Russell's paradox was it's an interesting story, and logical types and Turing and things, and machine learning and deep learning. Now we have this cognitive systems approach to everything. What are the paradoxes that we're going to face? And maybe they're not seeable now, but why is this better in a sense?
Better?
Well, we went reductionist, and they tried to build up from there, and it fell apart on logical grounds. And
well,
now,
it just isn't the way it is.
Okay, right.
The problem with it isn't that it doesn't work. It's just not the way it is. Okay, that just isn't the way it is. And what I'm saying is that is that exactly that paradox applies to this simple seeming question of systems that regulate systems that don't regulate themselves, right? You can make something not regulate itself by an act of law, or it can just be not able to regulate itself for other reasons. For example, if you order me to not have an accent, I mean, you can tell me that, but I don't know how to regulate. I mean, it's almost impossible if you have an accent to not have an accent. Is it right? I mean, it's almost impossible, and this is, it's a matter of not being able to regulate yourself, right? It's a regulation problem of a fairly simple kind, but I mean, well, it's not simple at all, but I mean, it's a very restricted example. But the point isn't whether or not it's better. The point is whether or not it is the way it is or not, right? So what this gives rise to is notions of authority. Like designing authority systems, you can't get away with saying, I'm an engineer, I don't care about how authority is distributed in this system. Or I'm a scientist, and that's not a scientific question, this is a political question or something. I mean, yeah, it's a political question, but in these systems, it appears. It appears, and I think that you can have a technological opinion whether or not, you know, you have a political opinion. Now, you have to have one, because you're going to design it in. You're going to have to design in, I'm not going to give examples, they take a while to warm up to give them, but there are issues of what do you mean by privacy, what do you mean by the publicity of a thing, right? And what do you mean by authority? Those things and others are inevitably essential components of design. You have to build it into the design. It's not possible not to build it into the design because that's Russell's paradox, and it's typed. There's no such thing as a level way to do this, right? It really is, that's the assertion. Not that it's better or something. Now, this assertion could be wrong, but that's the assertion.
Any other question? Yeah, go ahead.
Do you think the extended cognition that is brought by all this technological innovations has something to say about human rights and access to information, and do you think that those two will evolve together over time?
I think they inevitably do. I mean, I think it inevitably has something to do not just with human rights, but with just notions of, you know, what autonomy is and you know, what agency is and how it is reflected in authority systems. So yeah, I mean, if you go to Singapore, I really like Singapore. Singapore is an amazing place. You can walk around at 1 o'clock in the morning in the middle of this very large city in Asia, completely unmolested. I was in a taxi cab that rear-ended another taxi cab. Not too bad, but I mean, definitely ran into him. The drivers got out, they got on their cell phones, and I was, you know, listening. They turned around and looked because there was a camera. They were interacting with their cell phones and looking at the camera. A policeman never showed up. Some guy was assigned, you know, blame. Another guy was to say the thing was filed. No policeman showed up. The new houses that are being built there, apparently, I don't know this firsthand, I know this secondhand, but I heard it from a presentation. So unless the guy was not telling the truth in the presentation, this is at least reflective of something. In the new housing that's being built in Singapore, and 90% of people in Singapore own their houses. That's a whole different story, but it's related. Because own is a different thing, maybe. If the police have to show up at your house, or the firemen or something, they're going to know who's in the house, where they are, everything. The houses are completely instrumented. And they think it makes them a lot safer. So there's a big question in that. Like, it does in a way, right? So how that gets resolved is a matter of how technology and knowledge and the ability to know and who has the right to know, and authority systems and privacy and publicity interact. It's inherent to the design of the system itself. Its purposefulness, what you're regulating, who's not regulating, and so on, what an agent is, how you recognize them when they appear, all of the questions I just said appear in situations like that. And this isn't the only place in the world this is an issue. It's all over the place, obviously. And I think, yeah, so that's the point, actually. That's the point. The point is that we're, if, you know, so I have cartoon-like stories I tell of this. So if my theory of you is whatever's inside your shirt, and I have this method, and I say, well, it's not exactly true, but it's really close. This is an approximation. I'm a physicist. So I have a test to find out if I'm dealing with you or not, and that's to throw darts at the thing in your shirt, right? So if I hit the thing in your shirt and you squeak, I go, yeah, okay, well, that was you. If it doesn't hit, then it's not squeaking. I go, okay, so that's my theory of you. And then I say, okay, where's your money? Where's the process that pays your debts with your money? Where's your debts? So is that you? Well, if I turn off the process that pays your debts with your money, we'll find out that that's part of your autonomy, right? It's part of your peripheral self. And it can be really smart. It can be, you know, can have all kinds of features and stuff. So we are distributed. We're getting more distributed. We actually have agentic responsibilities to these things. They have agentic responsibilities to us. It's not at all as simple as what's in your shirt is you. It just isn't true. And the systems we build are changing that in a way that we don't really have a handle on. And I think it's extremely important that we do. And I think that there's a biological perspective of it, and there's a sociological perspective of it, and there's this junction between what it is that you do that's determined externally to you and what you do that's determined in a mediated way internally to you. Because that's what's being manipulated. And yeah, so that's the answer to the question is, yes, I do think that these things co-evolve. They are co-evolving.
Great. Thanks, Chris, once again. So last part of this symposium.